So every week I try to bring different books, and so they're up here on this table that relate to the topic we're discussing. So you'll have some references there if you'd like to look at some of those things to help out. So last time we were talking about, of course, in apologetics, we were talking about different aspects. This last time we started in the life sciences, because I feel like that's one of our strongest areas to look at. It's a very favorable area for helping us to understand the evidences for God. And again, in the whole series, some of you may or may not have been here. In apologetics, of course, we're making a defense about our faith and why we believe what we believe, so we can give a reason, a rational reason for what we believe. But also, there's an effort for offense as well, to go on the offense of, attacking ideas that are false, and then a proof, giving evidence, a proof of our actions and why we believe what we believe. And we were looking at the different areas. We looked at the philosophical evidences. That's a strong area, and we explored that. And then we talked about that there's scientific evidences, and then there's going to be historical, and that'll be what we look at next week, of looking at the evidences about Jesus' resurrection and about the validity or proof of accuracy of the New Testament documents. We talked about historical criticism and things like that. So these are some of the areas that we're focusing on. Now, this week, again, we're continuing with the scientific evidences, and I feel like that we looked at, the different areas that have to do with physics, I guess it was, and we talked a lot about my times with Dr. Guthrie over here, and there's so much about the physics and just the fine-tuning of the universe. There's a whole body of evidence about that that's just fascinating. But I find even more fascinating when we get into the biological realm of just the strong evidence for design and how this is just such great proof about why we believe what we believe. So there's been so much done. I don't know why that slide came up. Well, that was when we had it first. So anyway, that was up there because, again, if you've seen the movie Contact, there's some lines in it that are just really speak a lot to what we're talking about. And one of the lines is, "This looks engineered." And as you remember, the main star, Ellie, had spent her whole life looking for something out there that might show there's intelligence out there. And then the other person in the control room says, "Whatever it is, it's not local." And of course, that's the case. That's really is what's going on when it comes to our faith. And what we're seeing in biology is because it really does look engineered. The more we've gotten to know it, we spent some time talking about Darwin, and back then, and there's a book here, it's called Darwin's Black Box. For him, the cell was just a glob of protoplasm. They knew there was a membrane there, and they could see the nucleus, but that's all they knew, and they didn't really know of any significance of what was going on there. And so there's so much more now that we know. And so that's the teleological argument in philosophy, and of course that has to do with design, and we see that in nature, and we have these verses that speak to that "I will praise you, for I am fearfully and wonderfully made. Marvelous are your works, and that my soul knows very well. For every house is built by someone, but he who has built all things is God. And for since the creation of the world, God's invisible qualities, his eternal power and divine nature have been clearly seen, being understood from what has been made, so that people are without excuse." So this looks designed, as we said in the movie. And some of the characteristics when you're talking about design, we've hinted at those when we've looked at the other things previously. But the idea that it's improbable that it would have occurred otherwise than by design, that it has familiarity of what we think of when something is designed. It has this sophistication or intricacy about it that speaks of design. It has informational dependency. It's goal-directed. It's not naturally explainable. It's efficient, irreducible, complex decision-making and reflection of choice in the process, like engineering would be. So those are some things that we look at that are hints, and we see. So here's just a brief thing about the cell. Like I said, Darwin didn't have any of this information of all these different complicated features that are in the cell, and we'll look at some of that. But one of the things that stands out that's become more and more apparent in more recent years is these molecular machines that are inside the cell and then affect other aspects of the organism. And so there's some different examples, and we don't have time to look at all of these, but there's some interesting ones that we'll look at and we want to examine. But and we don't really have time to go into DNA, although that's one of the most fascinating ones, but we wouldn't have time to cover all of these things. But some of the aspects with DNA to keep in mind is that it also requires very complex molecular machines to make it work. And then that'd be some of these things like here, the DNA polymerase, and then these are very interesting here. And you can see some of these if you go online, but the helicase is, of course, the one that splits the DNA apart so that it's opened up. And then the topoisomerase, I was just thinking about what an interesting example. We were getting this all ready this morning. The wires, they were all clumped together, and you can't believe how always you get a wire or a cord, anything, that always gets tangled up. Well, the same thing happens with DNA. You think about this gigantic molecule that's really, I guess, about the biggest molecule ever seen, over 3,200,000,000 nucleotide segments, and you've got this stretched out. Well, it can get all kinds of tangled up. And so that's what this topoisomerase does that is up here. It can unwind that. It's amazing. All these machines are inside the cell working all the time, unbeknownst to Darwin or so many others that don't believe that this thing's a design. Very complicated processes that have to go on in mitosis, and then when you're trying to replicate and make more cells, or in meiosis, of course, with the sexual reproduction. But all of these things have to be kept straight or they get very glumped up. And then it's all pulled together with these machines to pull it together to store it when it's not needing to be translated or activated. So there's tons of these different molecular machines active at all times. So it's a very amazing, complicated process. So this gets to that first one I had listed there about the molecular machines involved with cilia or flagella. They have a very complicated process of how it makes these machines work. And that just gives you some examples. They're turning or twisting. The cilia, of course, are very important. We think about that with all this respiratory illnesses with COVID. It helps to clean out your lungs. They pull things up through in one direction to get it out of your body. And so these require a motor. And so this is just an example of one of those, and there's all kinds of illustrations where you can actually see it turning and twisting. But it's a very typical of what a motor would be. It's got the stator. For your engineers, of course, know all about this, and the rotor down there at the base that we're looking at, and then it turns and twists, and of course it's powered by the energy unit in the cell, which is ATP, and pushing hydrogen ions through protons. As they go through, it makes it turn. And so this thing is amazing. Like in the bacterial flagellum, for instance, they have clocked the RPMs up to 1,000 RPMs, so it's very fast. And it can switch directions instantaneously almost. So it turns the opposite way if the organism needs to turn. So it has an instant response, almost 100% efficiency of a motor to be able to do that. It continuously runs, and it has a clutch. So since it's continuously running, it has to have a clutch to release it so it can stop if the bacteria doesn't need to be moving. So just an amazingly complicated motor. And when you get to the idea of irreducible complexity, in other words, there's not any part, if you take it out, it's going to work. So how do you get a stepwise evolution like Darwin was talking about? He says if there was anything that required different parts for it to evolve and it couldn't function, then that totally dispels my theory. He said that himself, as we read last week from his direct quote. And so there's an example. And there's tons of these examples of these molecular machines. Now, there's all these other things that go on inside our bodies that's really like a machine in a way because you've got this very complicated chemical reactions that have to go on, like we mentioned before last time, that without enzymes, they're too slow to do anything of any good. So they have to have enzymes. So all of these steps, the main purpose I'm trying to show here is that every one of these has an enzyme that's making this thing happen. And so without those enzymes, nothing happens. And of course, the fuel that we all depend on in human life is glucose. And so this is the breaking down of glucose to produce the energy that we need to make energy in the cell. And that's the mitochondria plays a big role in this. Now, glycolysis takes place outside in that diagram we have in the cell. It happens in the cytoplasm outside of the mitochondria. But inside the mitochondria, then you have the rest of that process for breaking down the glucose. So the first step after you get through that big equation we saw earlier of that process and formula, we had the pyruvate. You get two of those when you break down one glucose. So glucose is 6 carbon atoms, and you can see here there's 3 carbon atoms in a pyruvate. And then that has to be transferred into the mitochondria. So when it goes in there, it's going to release this high-energy compound here called NADH. On the other one, when we're talking about glycolysis, it ends up, the energy cost will go through that, but it produces 2 ATPs. And then, so inside the mitochondria, you have this process, which has got a number of different names, but the Krebs cycle, a lot of people call it that. And it produces more of these high-energy compounds, NADH. It also produces GTP, which is like ATP, essentially. And so you get these compounds being produced from one glucose. You get all of this going on. And so it's done by molecular machines as well. And this is just one of those machines where this process going on in the first step of the electron transport chain. And so it's acting like a machine in the sense that it takes in the, well, I don't need that going on here, sorry.
Electrons to the next step.
Sorry.
In the electron.
And so this machine is taking, in the first step there, it's bringing in the NADH that we talked about earlier, and it's able to convert it, release the protons, that's the hydrogen atom showing up right then, and it releases it then to another compound which looks like those creatures walking in the membrane there, and that's another type of carrier substrate that moves these electrons. So what's happening is you're taking electrons and protons that come from the energy compound, the NADH, to get this process to happen. And so this is what's happening then if you're trying to understand the energy process. So you've got that high energy compound at the top, and it's going through this step of electron transport, this complicated process through those different enzyme complexes. And you eventually end up with water. And as you saw, well, you probably didn't see it, but it went too fast. But anyway, in the Krebs cycle, you're releasing carbon dioxide. So those are your waste products. So you're going to have 3 carbon dioxides from the 3 carbon pyruvate. So you're releasing carbon dioxide and water is being formed. But in that process, you're releasing these hydrogen ions and the electrons. And so there's another machine then that comes into play, which is the ATPase, which is going to create ATP from all of these electron and proton things that we've been talking about. And so it will create, and you can see again, this is these are large protein enzyme complexes is what it is basically. And so you see again in this one, just like with the flagella, there's a motor in there and it's turning. That rotation is what transfers the protons through, and in the process, it's going to create ATP. So this very complicated motor process going on inside the cell is making this happen. Let's see. So this is just more of a cartoon type demonstration of what we just looked at. You start on this side, this is inside of the mitochondria, and you've got these chemicals going through these processes in there. In each of these steps, it's transferring electrons, and ultimately it's able to break oxygen into and form hydrogen, water. And so then you're building up all these hydrogen or protons, if you want to call it that, on this part between the outer wall of the mitochondria and this little inner membrane space. So you get a whole bunch of protons here, building up pressure of protons on the charge. And as they go through this other machine, the ATPase synthase, it produces ATP. And ATP is the final product that we all depend on, all of life, from the smallest cell to the human body, in order for us to live. That's our energy source. And so here you get sort of the energy summary, if you want to look at it, is that from the glycolysis, that's the step that was outside in the cytoplasm. You end up, it costs 2 ATPs to make it work. So you only get 2 AT net and you get 2 NADHs. And then the Krebs cycle is the one that's inside the mitochondria, and it's producing these things. And then from that, the electron transport with the synthase, then you end up with that. So therefore, the final bottom line is 1 glucose molecule yields 38 ATPs. And so there's just millions of this going on in all the mitochondria producing ATP all the time, because every single transaction in the cell, all the molecular motors depend on ATP. So this is just a demonstration of, like in the human body, just an estimated of 200 to 300 moles of ATP per day is used. 1 mole is equal to 6 to the 10 to the 23rd power of molecules of ATP. But when you finally multiply that out with how many is needed, then that's the amount that's going to actually turn out to be up to 1.8 times 10 to the 26th power of ATPs per day. So we require a lot of ATPs to keep going. This is another molecular motor that's very interesting. All throughout the cell, there's a network or a skeleton of these fibers or filaments throughout the cell, and these different things in the cell have to be moved around. They're not just floating around crazily like used to be thought. They're all going to specific locations for specific tasks. And so you have these molecular motors that will move different things around. Like this is moving a vesicle down one of these microtubules, and it does it through the action of myosin, actin type thing. This one's kinesin though, but it takes ATP with each step. It's using an ATP to move it forward, and it'll move through these different channels, taking things to different places in the cell. Let's see. So then there's the process we mentioned. We just briefly mentioned about DNA replication, and that's a very complicated process, we said. But then there's forming the proteins, which are what is necessary for enzymes. So this is what we're talking about here, and that's another very complicated process. Again, so the DNA has to be split open. The helicase has to open it up, and then it's going to be copied. And that's where you get the messenger RNA, which then will go out to another very complicated molecular machine called the ribosome, which consists of various enzymes that come together to translate off of the messenger RNA and make a protein. Okay, so then that's the translation process that goes on. And this is just an example of that machine, of what it's doing. So it's copying. So each of those are these are transfer RNA bringing in an amino acid, and they're being put off of the chain of the copying from the messenger RNA, and it's making these proteins, and it's spewing them out of the ribosome, which was the red color there coming at the top. That's a protein being produced. So that's another molecular machine, and of course it requires a lot of ATPs to work. And then, so once you get the protein, though, a lot of things can happen. You can't just take a strand of amino acids together. That's a protein, which is great, but then it's not going to be doing anything if it's not. It has to be in a certain formation, a conformation, a shape, because all of these things depend on shapes to work. And different things happen. So they discovered what's called heat shock proteins, because what happens a lot of times, ultraviolet light, or in this case, heat, fever, whatever, it damages proteins, makes them denature, breaks them down. And so something has to be done. And so there's a process of trying to either repair that protein, or if it cannot be repaired, it gets marked for destruction, and they get rid of it. And so it goes to another machine, which is a proteasome, that destroys proteins that are not functional, that can't work. They're marked to say, okay, this needs to be discarded. It goes to the trash bin, or we can do something with it and repair it. And so the repair process is also part of the synthetic process. These are called chaperones. And so there's a number of different kinds of chaperones, which are type of heat shock proteins. But basically what they're doing is they're taking a protein like the one that just came off the ribosome and helping to get it in the right shape, because it has to have a certain shape to function. And there's a lot of different ways it could form. And so these different chaperone proteins are helping it to shape up to the right folding pattern. And there's another kind, these are very interesting to me, which they actually act like a special container for it to go in, and then the ATP is working with the enzymes in there to make that get into a folded pattern. And that's particularly important in certain ones that have a lot of very hydrophilic, because in the cytoplasm there's a lot of water, and that'll really mess up the way of the folding. And so this is a protected environment where it can be folded instead of by this other method that's outside in the cytoplasm for certain types of proteins to make that work. So it goes inside and has that where it can be shaped up right. There's a lot of different ones, but this is just some samples of it where the protein that's been produced by the ribosome goes inside, and then it gets closed up in there, and it's able to be folded, and then it's eventually released so it can go about and do the function it needs to be. I think we'll skip that. We don't have a lot of time, but that's just an illustration of it. Let me turn that off. So here's the real thing, though, is this catch-22, which, again, is the problem. It really does get back to the chicken and egg. That actually is a good illustration, because you've got to have proteins in order to make the ribosome machine. Okay, so proteins are required for all kinds of things, obviously. And then, but you've got to have ribosome machines to create proteins. So that's a real problem. And how would you ever evolve that? And so, of course, one of the things that we mentioned last time about epigenetics, but there's more to it than that. There's also cytoplasmic genetics in a way. And when you realize, of course, understand that in order to form a new human being, which, of course, that's a big controversy today, you know, if you don't believe in that life is formed by the fusion of a sperm and an egg, but anyway, a new human being, human life is created when that happens. And so all of that material for the cell comes from the mother. The egg is very large. The sperm has almost nothing to contribute other than its portion of the DNA. And so that's where you get all this. In other words, that's where you're going to get the ribosomes already. And you're going to get all this other extra non-genetic material in the cytoplasm. There's a whole bunch of, I mean, the DNA is huge, but it's not enough to do all the things that go on in a cell. And so that comes from the cytoplasm that the mother is transferring to that new life. And again, that makes sense from the Bible that, you know, that there had to be this beginning. You couldn't have just a cell pop up and say, okay, I don't have any ribosomes. I don't have any problem. Well, how am I going to do that? And so that's, you know, critical to our understanding of what's going on here. Now, let me see here a minute. All right. So we want to look at another molecular machine that has to do with chlorophyll. And this just to give you a brief overview of the leaf, and you see that it has the different cells there inside. And those little dots, of course, are chloroplasts. And inside of each chloroplast, then you have the granum, and then you have the thylakoid spaces there that are shown on the over here. So that's inside of all these cells in the leaf. And, well, maybe we have time for this little brief thing, but this just shows going inside of a chloroplast. But so it has some similarities then to what goes on in a mitochondria. But now plants have mitochondria as well, because that's how they're going to produce their energy from glucose. But so here's all these stacks of thylakoids. And then you go inside of them, of course, as you see, it's a membrane. And the membrane, all membranes are full of all these different kinds of machines and different receptors. And this is just a demonstration of those. In this case, it's the machines having to do with photosynthesis and producing its products there, as you see. And they're turning, that cranking of that motor. Now, that same principle works for photosynthesis, so it has these little motors going on here. All right. Whoops. So, and the other thing that's interesting about it is that the different inside of there of the chloroplast, and then you have these photosynthetic areas here, like this particular one. It has antenna, so it's amplifying. It's again using that principle of the electron transport. So they're picking up light in more than one place. That just shows one, and transferring it eventually to this primary reactive site. And then this is inside then of what goes on inside of the chloroplast, and then with the photosynthetic center here. So you've got the electron transport system that we mentioned right here, and then it's going to create these protons. So again, you're building up protons inside of the thylakoid lumen. So that's going on, and you're getting the hydrogen ion transport. All of these are requiring the electrons moving through. You have the second one, which for some reason they're always backward, but that's photosynthetic system 2. And so each of these are producing, that one produces an NADH, and then you got your ATP. That's the same kind of ATPase synthase that we already looked at. So all of that is going on to produce ATP and NADH for energy use. And then that has to be used also in the chloroplast then to eventually, the goal is to create some glucose, which is the cell, the plant needs that glucose there. And so it uses that, transfers it through taking CO2 and converting it to glucose. So you get water in, and you get oxygen out, and you get CO2 in, and you get glucose out. So an amazing process, lots of energy, very complicated machine. This Calvin cycle, again, just to give you an idea of the complexity of it, that didn't transfer too well. But anyway, it requires all these different enzymes to make it work, and it's producing energy units of ATP or NADH. Again, these can be used by the cell for its energy. And that just shows the energy cost. And so just to remember, we got 38 ATPs from glucose, but notice how much it takes to make glucose. And of course, all of this comes from plants. That's where it all starts, is in plants. And you notice what the energy cost there, what their productivity is. So it actually ends up, in order to make a glucose in the plant, it required a total of 54 ATPs to make a glucose. And remember, we get 36 when you actually burn it, when you're getting the energy from it. So the plants, of course, they've got a lot of energy because they're using the sun. So they're not particularly worried that it takes 54 ATPs to make a glucose. Now, hemoglobin is very important, obviously, and I want to look at that for a minute. And this is just how you create hemoglobin. Again, it's going to be a complicated process requiring enzymes to make this all happen and eventually produce hemoglobin. They're spreading, doing it the British way there, I see. But and then there's this similarity. So there's a similar process for creating chlorophyll, which is very similar to hemoglobin, you'll notice, but it uses magnesium in the core of it rather than iron. And that's a mistake there at the bottom. That's heme, not hemoglobin. Should be. So that's just, I give you an idea of the actual protein structure. You see all the, around it. So what happens with hemoglobin, so you've got your heme in the middle, but your heme rather in each of these subunits, but these are all folded around, are globulin, big protein structures that surround each heme. There's four of them. With 4 hemes that have 4 globulins that go with it. And then this is giving you the comparison then of what goes on in like just one of the photosystems for a cell, and it has the chlorophyll in its big structure. So all of these things have a big structure around them. Again, this is just demonstrating that a little better. You can see here the heme. So you have 4 hemes in each hemoglobin molecule, and it has these 4 different protein structures that support and hold it. And the significance of that, though, is how the heme works, because what happens is it has a relaxed state and it has a taut state, and that's what allows it to pick up oxygen and to release it into the tissues, and then to pick up CO2 and release it in the lungs and pick up more oxygen. And that's a machine, in other words. So it opens and closes. And this is just a, well, let's see here, quick demonstration of that. You have to watch it. It's a little subtle, but let's see here. So that's an oxygen, the red at the top, and when it moves in, there's actually change in the way these in the globulin here, that's a valine molecule, but it moves into different positions, so that affects how the hemoglobin works. That shows how it opens and closes there. And that ring, if you look, how it does. So, again, hemoglobin is acting like a molecular machine. And so what happens is, by having that machinery in place, these different effects, and this is showing how you get, when you have low pressure of oxygen versus high pressure of oxygen, it releases oxygen or it holds on to it. And different things will affect it, like the acidity or the presence of CO2, and this other molecule has a big impact. And then, of course, carbon monoxide really pushes it forward, and so it can't get oxygen when you have carbon monoxide, so it's holding on to it. And the other thing that's very interesting is the hemoglobin, the fetal hemoglobin, just an amazing process of what happens there is that, that when the fetus is inside the mother, it's got to be able to pull the oxygen from the mother. So the fetal hemoglobin is able to hold on to oxygen much stronger than the mother's blood. And so the baby is able to pull, the fetus is able to pull the oxygen out into it from the mother because it has the fetal hemoglobin. And of course, when it gets out, then that's got to be broken down. And that's when hemoglobin is broken down, it causes bilirubin, and that's why some babies have, jaundice temporarily, is because they are making all this bilirubin, and if they can't get rid of it fast enough, they'll become jaundice because they're creating adult hemoglobin and getting rid of the fetal hemoglobin. So I think we're way out of time. Everybody's starting to motion me, Joe, and they want to stand on their head. So, but like Dr. Smeltzer was saying in communion today, life is in the blood. It said, only you shall not eat flesh with life that is in the blood, Genesis. And then again, in Leviticus, it says that no one in Israel should eat blood. And then in Deuteronomy, only do not eat blood, for the blood is the life. And then Jesus said, though, verily, truly, I tell you, that unless you eat the flesh of the Son of Man and drink his blood, you have no life in you. Whoever eats my flesh and drinks my blood has eternal life, and I will raise them up at the last day. For my flesh is real food, and my blood is real drink. Whoever eats my flesh and drinks my blood remains in me, and I in them. Just as the living Father sent me, and I live because of the Father, so the one who feeds on me will live because of me. This is the bread that came down from heaven. Your ancestors ate manna and died, but whoever feeds on this bread will live forever. And so that's what it's all about, and we want to, there's a bit more on this biology. We obviously are out of time today, but we want to go next time, particularly in historical Christian evidences, as we start to look at the evidences for Jesus' resurrection.
I'm just sitting here listening, Mike, and you're doing an excellent job presenting multidisciplinary evidence of what's going on in science. But I've further observed that all of this is going on in each of us in a perfect manner. How many machines operate perfectly? Yes, they wear out, but they operate perfectly for many, many years. And people who believe in evolution will say, well, in 8 billion years, the chances are it can occur. But when they said that, they didn't realize how many enzymes are in the human body. And I'm sitting here looking at Googling, figuring out how many, and I saw a minimum of 1,300 to 75,000.
Mm-hmm.
And that would mean every one of them would have to line up exactly. That's what I have not been able to ever.
Right. Yeah, that's the truth. It's amazing. And there is some reason to even believe, and Mike and I have talked in the physics standpoint, where it talks about in the scriptures about that Jesus also holds everything together. There's some place there that there's more than we can even imagine of how all this can work. So it's a very fascinating, amazing thing, and hope you have a good week.
Very, very interesting to see the probability analysis. They would forecast what are the odds of any of them.
Oh, there's been a lot of those done. The numbers are so big it makes your head spin, but yes, there's a lot of those. All right, thank you.